Hi, and welcome back to our final section of Chapter 17, 17.5, Inflammation and Fever. These are two of the most important systemic responses of the innate immune system. We'll look at how inflammation starts, what mediators drive it, how chronic inflammation develops, and how fever enhances immune function. By the end of this video, you should be able to identify the 5 signs of inflammation and explain why they occur, explain the advantages and risks of inflammatory responses, and identify how fever starts and why it helps fight infection. Inflammation is a response to cell damage, stress, or pathogen entry. It begins with the release of chemical mediators, followed by the recruitment and activation of phagocytes. The purpose of inflammation, or purposes of inflammation, are to eliminate pathogens, remove dead or damaged cells, and begin the tissue repair process. However, if inflammation is excessive or uncontrolled, it can cause significant tissue damage and even become life-threatening. Acute inflammation begins immediately after injury. It starts with a brief vasoconstriction to prevent blood loss, followed by vasodilation caused by histamine release from mast cells. Vasodilation will increase blood flow to the area, so the area of infection, while increased permeability of the blood vessels allows the immune cells, proteins, and plasma to enter the tissues. And these early events help dilute toxins and bring in the components needed for microbial control. The 5 major signs of inflammation include erythema, which is redness due to the increased blood flow to the area, edema or swelling because of the fluids entering the tissue site. Heat is also due to the increased blood flow to the area. Pain occurs when there is swelling and compression on the nerves in that region. And then altered function. Altered function is usually caused by a combination of that pain and swelling, which can interfere with normal movement or activity. For example, pain will inhibit movement in that area to protect the injured area. The swelling usually also physically restricts the movement. And there's usually tissue damage in that region, which can reduce the tissue's normal capabilities. So for example, if you have inflammation in your muscle or joints, that usually leads to swelling and pain and limited movement and stiffness. And these signs are caused by that vasodilation and increased fluid leakage into the tissues, as well as the stimulation of those pain receptors, those nerve endings. And these images on the right show the histamine release from the mast cells and the phagocyte recruitment, all of which contribute to that redness, swelling, and warmth. The mast cells are amongst the first to detect injury in the tissues, and they release histamine, increasing the permeability of the vessels and allowing those phagocytes to enter the tissues. Phagocytes are activated, as we saw in the previous section, by pathogen-associated molecular patterns, those PAMPs. They're also activated by opsonins or distress signals from damaged cells. Complement protein C5a is also a strong recruiter that amplifies inflammation and attracts neutrophils to the area. Then several additional mediators will keep inflammation going. Bradykinin will maintain capillary dilation and contribute to persistent edema, that's swelling. Neutrophils are the first major phagocytes to arrive to that infected tissue area, and they will rapidly increase in number. Pus will develop as the neutrophils, microbes, and dead tissue accumulate, and we can see that here as well. Macrophages arrive later to clear this debris and allow tissue repair to begin. And this figure shows the progress of acute inflammation. In panel A, we saw that mast cells release histamine in response to injury. In panel B, we see the resulting vasodilation, so this blood vessel looks bigger, and there's increased permeability between those endothelial cells to allow proteins and phagocytes to flood the tissue. The resulting swelling, redness, and warmth all result from these vascular changes, and pain arises as the swelling and chemical mediators stimulate the nerve endings in that region. In contrast, chronic inflammation occurs when acute inflammation fails to eliminate the pathogen. The response becomes a persistent, lower level battle in our deeper tissues. But in chronic inflammation, this is basically a weaker, ongoing, drawn-out battle. We'll see mostly macrophages and lymphocytes, not neutrophils, and there will be overall lower intensity signaling and limited killing ability. Eventually, this leads to tissue damage and tissue repair happening at the same time, so the immune response is constantly active, but never reaches the intensity needed to clear that threat, to destroy the pathogen completely. And conditions associated with chronic inflammation include tuberculosis, hepatitis C, cirrhosis of the liver, and other degenerative diseases. Over time, the ongoing response leads to cumulative tissue damage. In some cases, chronic inflammation can lead to granuloma formation. These are pockets of infected tissue that are walled off by white blood cells. Macrophages attempt to eliminate the pathogens, but cannot fully do so. So this picture includes a micrograph of a tubercle caused by Mycobacterium tuberculosis, showing white blood cells surrounding an area infected with Mycobacterium tuberculosis. This first checkpoint asks, name the 5 signs of inflammation. So the 5 classic signs of inflammation are erythema, which is redness, edema, which is swelling, heat, pain, and altered function or loss of function. And these all result from that vasodilation, that increased vascular permeability, and the activation of the nerve endings. The second checkpoint question asks, is a granuloma an acute or chronic form of inflammation? Explain. So a granuloma is a form of chronic inflammation. Chronic over here. Granulomas form when the immune system cannot eliminate a persistent pathogen. So macrophages, lymphocytes, and other white blood cells will wall off the infected area to contain it, and this occurs over long periods, not during that rapid response of acute inflammation. An example is the one I just showed you in that picture, the tuberculosis tubercle found at that lung tissue. Certain parasitic infections can cause chronic inflammation with severe long-term consequences. Filariasis, for instance, involves nematodes, remember these are parasitic worms, that are residing in our lymphatic vessels, like we saw in the lab with Wuchereria. And this chronic inflammation leads to the formation of those granulomas. We might see fibrosis and also lymphatic blockage, so that blockage of the normal lymphatic flow. This produces elephantiasis with extreme edema as well as thickened skin, and secondary bacterial infections are common in these cases as well. Fever is a systemic inflammatory response that raises our overall body temperature. And body temperature is normally regulated by the hypothalamus. So here's the hypothalamus right above our pituitary gland. Fever is triggered by pyrogens, which can be exogenous like lipopolysaccharides found on gram-negative bacteria, or endogenous produced by our own cells, such as cytokines that are released by leukocytes, by white blood cells. So of those pyrogens that induce fever, remember again we have exogenous pyrogens from microbes, such as that lipopolysaccharide layer from our gram-negative bacteria. We have endogenous pyrogens that are made by our leukocytes, including interleukin-1, interleukin-6, interferon gamma, or tumor necrosis factor. And again, these endogenous ones are made by our white blood cells. These pyrogens stimulate the production of prostaglandins, which will reset the hypothalamic thermostat, and that will produce fever. So here again is my hypothalamus, and here's that little flow chart of how things happen. So exogenous pyrogens from microbes, induce the leukocytes to release endogenous pyrogens, and that will trigger the production of prostaglandins. Prostaglandins are made by endothelial cells of our blood vessels or cells within the hypothalamus as well as other tissues. These signals will lead to the hypothalamus to reset the temperature and cause fever. And fever can be beneficial. It offers several important benefits, including enhancing leukocyte killing. It also inhibits the growth of mesophilic pathogens because these pathogens prefer to grow best at around 35 degrees Celsius, which is equivalent to of around 95 degrees Fahrenheit. Fever also triggers the release of iron binding proteins or iron sequestering proteins. Remember, some microbes need iron to divide, so if you're essentially starving the microbes or depriving them of nutrients. A higher temperature also speeds up immune reactions. We're going to increase the rate of a lot of enzyme activity, and that will help the body fight infection more efficiently. During a fever, several physiological changes will occur. The skin might appear pale because in the extremities, you're going to see vasoconstriction of the blood vessels under the skin in order to divert blood flow away from the extremities, the arms, legs, and towards the core. You want the blood at the core to maintain higher temperature. Shivering will increase your heat production, and that core temperature will eventually rise. Eventually, the fever will break. Once the fever breaks, that's called the crisis phase, and you'll resume vasodilation of the extremities. So the blood vessels will dilate, and your blood flow will resume or move back to those arms and legs, hands and feet in order to release the heat and cool the body. Sweating will also help to cool the body and release the heat. While fever can be beneficial, extremely high fevers can be dangerous. Temperatures above 42 degrees Celsius or 108 degrees Fahrenheit can cause organ damage or be fatal. Here we're reaching temperatures where we start seeing protein denaturation or loss of membrane structure or DNA or RNA integrity. Some extreme fevers result from something we've seen before in chapter 15, superantigens and the superantigen-mediated cytokine storm. So these can be a result of toxic shock syndrome caused by Staphylococcus aureus or scarlet fever caused by Streptococcus pyogenes. This checkpoint asks, explain the difference between exogenous and endogenous pyrogens. So exogenous pyrogens originate outside of the body. They're produced by pathogens, for example, that lipopolysaccharide or LPS layer from gram-negative bacteria. And they're going to stimulate leukocytes to produce endogenous pyrogens. So endogenous pyrogens are made inside the body by our immune cells, like interleukin-1, interleukin-6, that tumor necrosis factor, or interferon gamma. These will signal the hypothalamus through prostaglandins to raise the body temperature. For the second question, they ask, how does a fever inhibit pathogens? So fever can help control infection by slowing microbial growth. Most human pathogens are mesophiles that grow best near our body temperatures, 37 degrees Celsius. That's normal temperature. Fever will also enhance leukocyte activity, so that will speed phagocytosis and immune cell recruitment. Fever can also reduce iron availability, so certain proteins released during fever will sequester or bind to iron, starving pathogens that require iron. And it'll also increase our metabolic rate to boost our overall immune response. The clinical focus example from this chapter highlights hereditary angioedema. And for the patient, what happened was that they were deficient in C1 inhibitor. So what that means is they basically did not have anything to inhibit complement activation, which led to uncontrolled complement activation. So the take-home message is it's great that we have complement proteins and we can trigger inflammation when necessary, but you don't want it to be uncontrolled. When it becomes uncontrolled, this leads to an excessive production of anaphylatoxins, and severe swelling. We can see it in these pictures over here. You'll see swelling in the airway, the GI tract, the extremities, and treatment includes providing the patient with purified C1 inhibitor or other anti-inflammatory medications. Some key takeaways from this section: we saw acute inflammation is rapid and protective, but chronic inflammation is persistent and damaging. Chronic inflammation can result in the formation of granulomas when pathogens resist elimination. Fever boosts immune function, but can be dangerous if excessive. And mediators like histamine, bradykinin, cytokines, and prostaglandin can drive both inflammation and fever responses. All right, let's recap 17.5. Identify the signs of inflammation and fever and explain why they occur. So let's look at the signs of inflammation first. There are 5 classic signs. Erythema, which was redness, was caused by vasodilation, which brings more blood to the area. The second was heat, and that's also due to that increased blood flow from vasodilation. The third one was edema or swelling, and that's due to increased vascular permeability, which allowed fluid, proteins, and leukocytes to leak into the tissues. Fourth, we had pain, so swelling and inflammatory mediators like bradykinin activated nerve endings. And the fifth one was altered function, so pain and swelling usually reduces mobility to protect that injured area. For fever, we saw signs of fever included elevated body temperature caused when pyrogens trigger prostaglandin release, and that will reset our hypothalamic thermostat. We'll also see chills or shivering to allow the body to attempt to reach the new temperature set point. Usually we'll see flushed skin and sweating, though, when the fever breaks. That was the crisis phase as the body's trying to cool itself back down. And these occur because of fever is a systemic inflammatory response and not a local response. The second learning objective asks, explain the advantages and risks posed by inflammatory responses. So for the advantages or benefits, we know that inflammation is protective and essential. It helps us eliminate pathogens. It brings those neutrophils and macrophages to the site. It contains the infection early, and it prevents the spread by clotting factors and swelling. It helps us remove damaged cells and debris, so those phagocytes will clear the dead cells and prepare for repair. And it also initiates tissue healing, so that increased blood flow will deliver oxygen and nutrients. Fever will also enhance immune function. It'll slow the pathogen growth, but speed up leukocyte action and also reduce iron availability. But the risks or harms is that there could be extensive or prolonged inflammation, like we saw in chronic inflammation that can lead to tissue damage. We could also see swelling in confined spaces that could be dangerous, like airway swelling that would cause airway obstruction, or brain swelling that could lead to increased intracranial pressure. And uncontrolled systemic inflammation can also be dangerous. Extremely high fevers can cause organ failure, and we saw that superantigen triggered cytokine storms like we see in toxic shock syndrome or other severe bacterial infections can be fatal. Other risks include chronic edema, so persistent inflammation like we saw in filariasis can also block lymphatic drainage and cause that constant swelling, that extreme swelling that we saw in lab, elephantiasis. And that takes us to the end of chapter 17. In the next chapter, chapter 18, we're going to look at adaptive specific host defenses. Thank you for watching.